So as we go up the value chain at this event, I am delighted to introduce to you an exceptional executive who was the first engineer hired by Kite Pharma exactly a decade ago, who committed himself to developing technology for clinical and commercial cell and gene therapy manufacturing. Ladies and gentlemen, that is no one other than Kenny Choi, who today is the Senior Director of Process Sciences and Engineering at Instill Bio. What makes Kenny very special in my heart is that he has served as a faculty member at the City of Hope Hospital, in addition to all the pioneering work he's been doing in what we have heard all morning is one of the most challenging areas of scaling up manufacturing. Kenny, it's all yours.
Devendra, thanks for the overtly generous introduction. I'm very happy to be here. In terms of presentation, were we still planning on you guys to present the deck for me?
Either way, if yours is handy, you might as well share, minimizes headaches.
Okay, well, I'll share on my end, and if we have some buffering issues, we can transition. Thank you.
Understand. Thank you, sir. Perfect.
Can everyone see the full-scale presentation?
Yes, sir.
Yep, thank you everyone for your time. My name is Kenny Choi. I'm currently Senior Director of Process Science and Engineering at Instill Bio. I'm here to present to you some of the high-level challenges that we face in process development when dealing with adoptive cell therapies. Quick overview of the agenda for today. I would like to give you a brief introduction on my organization, Instill Bio, in our focus area, our therapeutic focus area, which is TIL, or tumor-infiltrating lymphocytes. And then we can discuss some of the key challenges for PD when working with cell therapies, especially derived, from the patient. And then we can go over current gaps that exist in enabling technologies for this space, whether it's in capability or appropriate scale. And then we can discuss some topics about managing change and really differentiating continuous improvement for legacy products versus kind of pursuing self-disruption. And then I'll provide some closing statements. So Instill Bio, we're a clinical stage company, wholly focused on tumor-infiltrating lymphocytes in the autologous adoptive cell therapy space. We're also highly interested in engineered TILs to further enhance their cytotoxicity. We're based out of the greater Los Angeles area with some capabilities in Dallas and the UK. And we're currently building out our own internal, commercial, clinical, and process development infrastructure on both sides of the pond. Diving a little bit deeper on exactly why we're excited about TIL therapy. I think for the past couple decades, we've seen incredible progress and impact to patients when it comes to adoptive cell therapies. And this is mainly true in hematological malignancies, speaking to some of the engineered CAR-T and TCR products that we've seen come to market. Unfortunately for the patients, not a lot of progress has been made on solid tumors, which we really see as the high-hanging fruit in terms of patient need. And this is for several reasons. One, when we speak to solid tumors, we have to start considering the tumor microenvironment. This is not a bulk systemic blood disease. Second, product cells derived from your periphery won't have intrinsic ability to traffic, as in, they don't have the right tissue tropism for that disease. And then the most critical thing is targeting. When you have an engineered antigen, unless you go through some broad screening for kind of somatic mutations and design, against those neoantigens, you're typically targeting one or two tumor-associated antigens, and you have to come with a much stronger punch than that when you think about solid tumors. So really the clinical hypothesis is that if you find immune cells in tumor tissue, they must be there for a reason. That they're hitting all the challenges I just marked, but more importantly, there's relevant clones within that population that are reactive to that disease. And our idea is to kind of leverage the long history of clinical validation and further improve this approach in terms of harnessing kind of the inherent ability for TILs to target the cancer. And up until now, there's been a lot of progress across several indications, both in academia and industry, with very respectable objective response rates that we truly feel can be further improved. In terms of pipeline, we're focusing heavily on our optimized unmodified TIL programs. We are currently in the clinic for melanoma, which is our flagship, and in the early IND phase for CSS, non-small cell lung, head and neck, and cervical. And then we also have an upcoming basket trial focused on engineered T cells or engineered TIL. General end-to-end kind of manufacturing flow. This will be very familiar for those of you who are, engaged in CAR T or TCR therapies. A patient will check in to a clinical center. Instead of undergoing apheresis, in our setting, they undergo surgery where a sample of that patient's tumor is resected out, and that really becomes our starting material, both the tumor cells and the T cells in that starting material. We enzymatically and mechanically digest that starting material to create a single cell solution. We cryopreserve it. And then for the cell production, we have what we call stage 1 of outgrowth, which is really a rest in a cold culture setting between the TILs and the tumors to start providing stimulation and for them to come out of the state of total exhaustion in that microenvironment. Following a couple of days of that, we put them into REP, or polyclonal activation, or rapid expansion phase. And once we have a relevant dose, we cryopreserve the product. And following QC release, the product is thawed bedside and infused. On top of the cell product, the patients also undergo high-dose IL-2 therapy to support in vivo cell expansion. To highlight some of the differentiating factors of what we are doing. Historically, TIL products have been made from tumor fragments. So you have a larger bulk tumor fragment that is received as a fresh shipment. You manually or semi-manually fragment it into sub 1 centimeter components, and then you seed those tumor fragments, and from there, some of a portion of your T cell population or your TILs will emigrate out and start to proliferate in culture. With this, there has been characterization of what the field calls remnant TILs, and often the TILs that aren't salvaged and part of your product are the ones that are the most interesting in terms of reactivity. And it makes sense because if they are relevant to that tumor, they're going to want to stick with it in terms of tropism. So our approach, we've developed a highly effective automated process for rapid shipment digestion and cryopreservation of the starting material to really provide an operational pause, which helps us level load, but also maintains the attributes that we are interested in when it comes to both the starting tumor and the TIL population. Going back to our concept of the engineered TIL, classically, there is no gene modification or endowment of new functionality when it comes to TIL products. You're wholly reliant on the intrinsic or kind of native signal 1 through the endogenous TCR from that product. When you have poor T cell activation, there is measurable tumor growth or progression of disease. So, are there any learnings from the CAR T world that we can borrow, as in, can we engineer an artificial receptor that targets a tumor-associated antigen that, in our case, doesn't provide signal 1, but supplements the TILs with signal 2 and 3? And that's exactly what we plan to do. The construct that we plan to endow our TIL products we call CoSTAR, or co-stimulatory antigen receptor. We're still wholly reliant on the native TCR for signal 1, but through the tumor-associated antigen, we're supplementing with signal 2 and 3, so that we can enhance the overall functionality and proliferative capacity of our cells of interest. Let's dive into kind of more of the presentation. So I want to share with you, from what I feel like are some of the key technical challenges at each phase of development. And the way we're breaking this down from left to right really is kind of following, what the agencies typically define, as life cycle, stage 1 process design, stage 2 process qualification, stage 3 CPV, and how that really overlaps with, general cell therapy trials that enter registration enabling trials immediately as part of phase 2. So early on, it's really about feasibility or setting the stage. The first challenge I want to highlight is kind of sourcing indication-specific samples to support PD work. So, I think from our experience, if you're working, in heme and working with an engineered T-cell product, you can get quite far with healthy donor cells. If you're working on solid tumors, especially working in TILs, where you're interested in a very subset of a highly specialized, T cell population that has the ability to infiltrate the tissue and survive in the microenvironment, et cetera, you're going to want to base the body of your development work off of that indication-specific samples. So, it's quite challenging to establish internal biobanks, on top of all the other work, when it takes getting a company up on its feet. But you really need to take that into consideration as one of the more strategic goals in terms of, one, identifying your priorities, two, establishing a network of suppliers, both academic partnerships or commercial source, and then having some internal capability of kind of standardized methods of processing those samples and distributing. Second challenge, understanding variance from multiple sources. So, I think we're all familiar with this. In biologics, we've had to deal with this too, but it's even more true when you have to deal with an autologous cell therapy. For us, there's really 3 buckets. It's the patient starting material and the inherent variability we inherit there, the process and the analytical methods. And I'll kind of deep dive a little bit on this in the following slides. So let's move forward. Next item, selecting appropriate raw materials, consumables. One of the big challenges, for us is in general, we've been limited to, what we call catalog engineering. We're kind of Frankensteining manufacturing processes by borrowing things from, research use only, or, what's been established in blood banking, or, scaled down platforms from, traditional biopharma. All of that has to change if the industry needs to mature. So, being really mindful of, what materials you select up front, how wet are you? Is your process to those materials? What opportunities, are you salvaging for change and optimization in the future is quite critical. And then data plan. I think for all of us, we're in a new industry, so whatever therapy we're pushing forward, not only we should see the opportunity in approval, but using that as a platform for learning. And when you think about all the data being generated, in PD, in manufacturing, the translational, overall clinical aspect, and even the preclinical aspect of it, it's extremely challenging to piece that all together retrospectively, especially if you don't have a strategic data plan up front. So it's really something that you need to establish, when the company is young. Next phase, really thinking about refinement. So it's starting the process of establishing, product understanding. And I'll have a couple slides on this subsequently, so I won't go to make too many comments here. But of course, it has to do with characterization of your starting material, or process, and product, and tying that to the patient. And then clinical manufacturing experience. It's perhaps unfortunate, but you always learn more, in manufacturing than in PD. It's just how it goes with this type of work. So you have to be prepared, not only to face deviations and, address investigations, but kind of maximize learning in those situations. Next, implementing process improvements based on latest knowledge and capabilities. I do have a couple other slides on this later, so again, I'll move fast. but it's having a comparability strategy kind of mapped out up front, having that intent to improve the process, once you establish your initial IND for phase one. And of course, that's tied to, your overall process design, how flexible you've designed it, et cetera. And then enabling commercial scale out. So, if early on, the point is really feasibility. Once you're end of phase one and about to graduate into, pivotal, you have to be very serious about, what your operations look like in a commercial setting. The longer you wait during phase two, the more rigorous your comparability package has to become. But also because of the tempo, of the business, you lose opportunity. And kind of the general strategy for most companies is that the CMC timelines or the drug development timelines are never waiting for PD. So you have to catch that bus when you can. So late stage, maintain when we start thinking about commercial launch. Designing the appropriate, validation for your process and overall systems is critical. Because of the variability we have to live with, kind of this idea of PPQ for an autologous cell therapy is still not straightforward. If you follow kind of the traditional statistical approach and do power analysis, because of the variability, it's going to tell you to do 100 plus runs, and obviously that does not make sense both technically and from a business perspective. So you have to put a lot of thought into what criteria select. Have you identified the right criteria? During characterization, are you sure they're relevant to your product quality? And then if you're not able to satisfy kind of more of the traditional track when it comes to power, power of the study, how do you justify kind of lower sample size? And then some other things, I'll move fast for the sake of time, maturing control strategy and product attributes. I think this goes hand in hand with kind of maturation of your process. And it's the same concept of not tighten the screws early on when you're building furniture. There's a lot of learnings across the life cycle. So make sure you have flexibility in kind of adjusting your control strategy over time. Establishing material specifications for critical reagents. All the materials and consumables you use, there's a certain label on them that the vendor, the manufacturer will provide, research use only, GMP grade, clinical grade, pharma grade, et cetera. At the end of the day, the agency sees the sponsor, of the program accountable for confirming quality. So keep that in mind. You can use research use only, as long as you provide, the appropriate level of internal testing. You can use GMP grade and still require additional testing for more, specific criteria, for that material. Capturing end-to-end development story to support BLA filing. It's very challenging to do this piecemeal. When it comes time, for your application, it's an enormous story you have to tell. So start building it up front systematically. Establishing long-term viral vector supply for commercial scale out. This is even a challenge for phase one, but when you think about the total opportunity we have in cell therapies, especially in solid tumors, when we're starting to talk about greater than 10,000 patients in need a year, per any given indication, we currently don't have enough global global supply capacity when it comes to vectors. So if you're a company that uses viral vectors, plan for success and kind of map that out, whether you want to bridge using CMO, but intend to internalize some of those capabilities, but acknowledge the lead time in developing manufacturing and also bridging. And then really the concept of global change management. As your manufacturing network gets more and more complicated, you're not dealing with a single facility. It's international network of commercial plants. So this again kind of goes to the concept of flexibility. You're not going to be able to control for everything, for all of the site-specific needs or local needs. So understand what's critical in your process, allow for some flexibility and even the equipment you use. And of course, these are some things that can be addressed in tech transfer, but from validation perspective, it has to be tackled up front. Okay, kind of focusing on source of variability, what we deal with. In terms of relative variability, I think the biggest source is the starting material for us. So when we think about a solid tumor as your starting material, even when you're starting with an apheresis from a patient, there is some effect I call kind of central limit effect, where you have greater than millions of clones from that patient from the blood, and therefore a milliliter of blood will generally behave the same as a milliliter of blood from somewhere else or later in the collection phase. When it comes to a tumor, there's sub million clones often. So even when you design a perfect study, for a split study, you could have heterogeneity issues. So, all of that is kind of baked into the total variance you have to deal with. Manufacturing process, I consider it very low. I think a lot of us are kind of working on our Gen 3 and 4 processes. Early on when we first inherited these type of products from academia, it was very dizzying. But I think right now we have pretty good control. Analytical methods, most tests you have to rely on are cell-based. So think about what standards you follow, especially what controls you select for this. And then when it comes to ability to control, what we inherit from the patient is what it is. Baked into that, is the patient history, the state of the disease, and many other things that we just can't connect the dots on quite yet. So, I consider that low control, medium control for the process, especially early on. And then, you should be able to control, your analytical methods very high if you follow the right guidances. And to kind of reiterate that, how you control for these in development, for the starting material, really, there's nothing you can do until you earn the understanding. So, really think about maximizing data collection. Don't compartmentalize. Every feasibility study you do, every kind of at-scale confirmation or IND enabling run, a clinical runs, analyze that continuously in totality. And again, it's hard to do that retrospectively. Manufacturing process. I've said flexibility a lot, but keep it flexible. Before my presentation, someone had some really good comments in that, there's a lot we're learning from translational perspective when it comes to cell-based therapies. We haven't quite identified, what the key drivers are in terms of mechanism of action. There's some good hypotheses, but it's actually a very complex play, on things, far beyond kind of primary mutations and even some reliance on, your endogenous immune system. So the more you try to optimize up front, the more opportunities you lose out on in terms of learning. Let's move on for timing. Characterization, you can follow very traditional path, as you would in biologics. You have a collection of prior knowledge, you have clinical manufacturing experience. Piece that all together as part of a risk-based approach, understand where you need to pay the most attention to, do deep characterization in terms of operating range, edge of failure, and use all that knowledge to establish your validation. In terms of how you approach, process understanding, this is tied to the data piece again, but plan to throw a very wide net out early. There's a cost to it, but it's worth it. Over time, or earn your right to distill that down to more and more of the relevant questions. If you don't do this early on, it's much more costly to try to do it retrospectively. And then the technology or general scientific understanding is catching up. It's extremely exciting decade. So even if you don't know, exactly what the sample will be used for, what tests you'll run, what data, that data will be used for, we still should have a pretty good understanding of where in the process, the samples will be interesting. So, do that quite aggressively across, PD and clinical, and establish a really robust inventory of samples, because at some point you're going to want them. Some additional challenges, that are triggered, by early PD decisions, that are extremely challenging to adjust, because you measure them in a kind of at scale operational setting. And I apologize for not managing my time well, so let me go through just the issues here. But it's level loading, just capacity at your plant. There's you want to manage costs when it comes to direct labor. So the easier it is to schedule and predict your flow of labor, the more efficient that will be. Capacity as a function of space. So a lot of the technologies we rely on, are designed for the bench. it's really what academics have been doing to manufacture these products for a very long time. We have to graduate from that as an industry. So transitioning from batch or bench model to more of a ballroom concept, and in effect designing the technologies that could take advantage of that flow versus really thinking about the operations on a bench to bench per patient basis. Turnaround time, of course, that's tied to patient experience. And then critical reagents and consumables, avoid single points of failure. We never hold inventory of final product, so even the smallest kind of supply risk support manufacturing can be catastrophic, so you have to address those up front. I'm out of time. I'll take 2 more minutes. I'll go really fast. I want to highlight kind of technology gaps. The needs are vast, and the technologies have not caught up yet. And there's many reasons for this. Systems engineering is, the principles of system engineering is very prevalent in many other disciplines, not so much in biopharma. And I think I truly feel that's because the margins have been so forgiving. We see that that's not the case for autologous adoptive cell therapies. We have to become much more rigorous in terms of not just understanding how our process behaves on the bench with human hands, but how the design of the process impacts the flow of data, material, product, labor, across the plant and across the network down the road, and you have to think about that up front. Fault tolerant engineering, when we have a patient's material in our hands, there's no difference in terms of risk from that patient being on an operating table. We have a lot of equipment where we're completely okay with 1 out of 20 failure. From my perspective, as this industry matures, that should be unacceptable. There's already well-established kind of verification, validation principles that we can borrow from other industries to make our technologies and processes more robust. And then, of course, material science. We're still borrowing, again, like I said earlier, from blood processing, traditional biopharma, using a lot of the plastics in terms of biocompatibility that have been established there. There's a lot of other capabilities that we can start bringing in-house, and that have already demonstrated manufacturability. And I'm hoping we'll see more of that. This is speaking more to kind of the systems engineering. Let me fast forward to kind of the take home here. Critical priorities for early stage PD. Establish a data plan. Get rid of all the silos. Establish a central data lake, map out where all the connectivity is, and be very serious in terms of investment there, so that your first programs really can be treated as a platform for learning. Be very ambitious in terms of the amount of questions you ask up front, and again, earn your right to distill down to the important ones. Keep the process flexible to address the variability and to maximize learning. And then, as you design your process, consider systems level implications. Again, it's really about the end goal. We're not here to support, just clinical trials, but we want to see success of this industry in a commercial setting. So think about what drives COGS, at that scale, and then, be really intentional about self-disruption. Quite often, because our development timelines are so aggressive, what is lost is kind of the foundational biological questions, which should precede your engineering problems. Because of that, we lose out on opportunities to kind of go a little bit beyond improvement and really think about innovation. So, push forward your programs, start improving them, start making them, more robust in terms of closure, simplification, reduction of materials or secondary sourcing. Parallel to that, very aggressively establish, what you should consider your Gen 2 manufacturing process. Completely different from what you're doing. Don't be afraid to, dedicate a clinical cohort to it, and don't limit yourself to the questions you could potentially ask, as part of some analytical comparability package. Again, I apologize for not managing my time well, but I'll stop here for questions. Thank you again.
So Kenny, very honestly, I'm amazed, and if I may say so, you are our rock star for manufacturing process development in cell and gene therapy, and we wish you the very best. And thank you for defining what is such a complex and difficult technology. Hats off to you.
Again, thanks for the overtly generous feedback, and it's a team sport. So I think none of us want to be, captain of a sinking ship. So, let's all work together and mature this industry together. Thank you.
You certainly will. And thank you again. And we'll move on, and we will find other avenues to work with you and ask you a lot more questions. Thank you.
Cheers. Thank you for your time, everyone.